Okay, we are going to start Chapter 5 Notes, Part 2, which is talking about transport across membranes. And I've got some stuff pre-written here, so I can make this a shorter recording and you can pause if needed. First of all, just change Chapter 5 in the Barron's book that you can review. And we're going to talk about transport, how molecules move into or out of cells across membranes in general. But that's going to include not just how they move in and out of cells, but just kind of general molecular motion. So you'll see here first, we have two types of transport: passive transport and active transport. Passive transport is a negative delta G, which if you remember what that means it's not requiring energy, it's actually releasing energy. It's like a downhill reaction. Active transport is going to require energy. That's what a positive delta G means. Okay, so generally, when molecules move from a high to low concentration, which we say with their concentration gradient or down their concentration gradient, this happens naturally just due to random motion of molecules. Things will spread out evenly. So if molecules are going from high to low, whether that's across the cell membrane or just in a system, that is not going to require with energy. So this happens without the use of energy, or ATP. So I wrote that up here too. This is what happens, passive transport, this is what happens naturally just because of random molecular movement. Molecules will just tend to spread out equally, and so they'll tend to move from areas of high to low concentration. So the examples of that we talked about freshman year. There's basically diffusion and osmosis. There's really two. Diffusion is when the solute moves from high to low. And then we're going to see facilitated diffusion is just diffusion through a protein, but it's still diffusion. Osmosis is when water moves, which is the solvent, is going to diffuse from high to low. And aquaporins are just proteins that make that happen faster. So we have diffusion by itself, diffusion through a protein, osmosis by itself, osmosis through a protein. And I'm going to address all of these here in the notes. This is kind of an overview of everything we're going to talk about. Active transport requires the use of energy because it's molecules moving against their gradient. So they're moving from low to high. Think of that like an uphill. So that we could also say they're moving against their concentration gradient or up their concentration gradient. This will not happen by itself. So if a cell wants to move something from a low concentrated area to where it's high concentrated, it's going to have to use energy in some capacity. So we're going to see a lot of times that could be ATP is used as the energy source. Or there's also something called coupling or co-transport, just like we talked about coupling of a positive and a negative delta G reaction. We can sometimes, without using ATP, use something that's moving passively from high to low can kind of provide energy to move something that's moving actively from low to high. And again, we're going to go in all details of this. I just want you to get in your head right now kind of the general differences. These are just some examples. Pumps, this says sodium potassium pump. That's just an example, the sodium potassium pump that we will do. But pumps in general are, when you hear that word pump, it means it's a protein in the membrane that is pumping something from low to high. It kind of implies active transport. So I also wrote up here for you to add in active transport. Because things are moving low to high, it's going to require energy. But it also usually requires a protein in the membrane to either do the coupling or have ATP bind to it or something like that, which you will see. And then we'll talk about endo and exocytosis when the membranes pinch in and out. That requires energy, so we've put it under here. This notes that we're starting today, I'm going to primarily kind of just address passive transport first, and we're just going to go over some basic stuff. So I'm going to talk about all of this, okay? Just some terms to know what a concentration gradient, that means there's a difference in concentrations. So up here, when I talk about moving with its gradient or a against its gradient, that means there's a difference. If the concentrations are the same on both sides or in all areas, there's no gradient. So there's going to be no movement overall of molecules. Equilibrium is the term we use when concentrations are equal everywhere. So you either have one or the other. There's either a difference in concentration or there's not. Okay. Solute, just to remind you, is the thing that's being dissolved in a solvent. In our situations, remember, water is always pretty much a solvent in living things. So looking up here at diffusion and osmosis, they're both the same thing. They're both molecules moving from high to low. But when the solute does it, we call it diffusion. When the solvent, water, does it, we call it osmosis, which I wrote right here. Diffusion is the movement of the solute from high concentration to low concentration. Osmosis is the movement of the solvent, water, from high, don't say high concentration to low concentration. That's confusing because concentration kind of involves the solute, okay? When we define concentrations of a solution, we are usually giving you the concentration of the solute. So just be careful. Water is going to be where there's more water to less water. That's going to be osmosis. And you could have these happening kind of at the same time. They tend to go in opposite directions if the membrane is permeable. So let me switch over and just give you kind of an example that we sort of, I think we did something like this freshman year. Let's suppose I have this hypothetical beaker with a membrane down the middle, and let's assume that the membrane is permeable to both, let's assume for this example, it's permeable, the membrane, to salt and water molecules. Okay, that's going to be the key. So let's say side A, I have a 10% salt solution, and side B, I have a 30% salt solution. So if I asked you to show me diffusion with a red arrow, okay, which way is diffusion going to go? Remember, diffusion is talking about the solute. So I have 30% salt on that side. So my overall movement of salt is going to be going from B to A. And we generally write it like this because some molecules move from A to B. There's salt molecules just over here bouncing around with kinetic energy. There's more here, okay, if those are salt in red. So the molecules are, they don't have brains. They're not like, hey, you guys, we're only supposed to move from high to low. A few of these salt molecules might bounce this direction and cross the other way, but more are going to go this direction because just there are more salt molecules on this side. Just based on probability, more of these will randomly bounce that way. So we would say the net, we kind of refer to this as sort of like the net movement of diffusion is this big arrow. Okay, so overall, salt is going to be going from high to low, 30 to 10, that way. Osmosis then, so they don't give you the osmosis. There's not like a water concentration. That's not really the term we use. But if it's a 10% salt solution, what that means is that 90% of the molecules on this side are water. And then here, that means 70% of the molecules on this side are water, right? Because it has to add up to 100. So now if I'm asking you for the direction of osmosis, let me make this a little thicker. Okay, water is going to go from where there's high water to low water by osmosis. Again, a few water molecules will go that way, but the net movement will be from side A to side B. Okay, so remember, this is the solvent. Osmosis is the solvent. It's water. So we wouldn't say water's moving from a high concentration to a low concentration because concentration kind of implies the solute number. Okay. And neither of those are going to take energy. These, as long as the membrane is permeable and molecules are allowed to cross, this will happen until everything is equal on both sides, and then it will be at equilibrium. The molecules will still move at equilibrium, but then the arrow going in both directions would be equal. It's kind of, there's no net movement in one direction or the other. Okay. So that in general is diffusion, osmosis. A couple other things I want to, so here's a definition of diffusion, okay? It's the net, which just means overall, like I showed with the two arrows, movement of molecules from high to low. It's due to the random movement of molecules, remember kinetic energy. It's the movement of solutes. And then at the end, we call it equilibrium. So here's a similar thing. If the red molecules are the solute and the blue molecules are your water, your solvent, I would say right here, I have a concentration gradient in this beaker. There's a higher amount here than here. Diffusion is going to happen. The red molecules are going to randomly move and spread in this direction until they're all equally spread. And then I have equilibrium. So this just shows you, I kind of showed you what I mean by the net, I wrote how to draw the arrows for that. So I'm going to give you you might want to take a second and pause and write this down, but let's say this pink line here is a membrane. Okay, so we're going to talk about two sides of a membrane, usually, not just in a beaker, when we're talking about cells. So let's say I have a high concentration of sodium, right? The brackets just mean concentration, on side A, and I have a low concentration of sodium on side B. Diffusion of sodium would be from A to B. Okay, the net movement would, but remember, a few might move that way, but overall, I would say diffusion is happening from A to B. It's just kind of how you show that with the two arrows. Over here, I've listed some things that can affect the rate of diffusion. Okay, most of these things, I think, are things that would speed up diffusion. So when the molecules are smaller, they tend to diffuse faster. They tend to have like more kinetic energy, more random motion. So it tends to happen faster. Temperature is fast. At higher temperatures, it's faster because, right, heat is energy. Heat is basically adding energy. So if I heat something up, remember you have a higher kinetic energy of the molecules, and so they're going to tend to just spread out faster, okay, at high. Like if I have hot tea and I'm trying to dissolve sugar, it happens faster than in iced tea. So the concentration gradient, meaning the difference, if you have a steeper gradient, so if I'm going from 90 to 50, concentration, that's going to be faster than if I'm going from 60 to 50. Okay. Charge. So the main ones I want to talk about here, an example. Charge. Faster towards the opposite charge. Okay. So let's suppose I kind of did this in blue here. I'm dealing with, again, this membrane, and I have high sodium on this side, high concentration of sodium on side A, and I have a lower concentration of sodium on side B. But let's also suppose that side B has something else that has negative charges. Okay. So if side B has a negative charge, sodium, it's going to diffuse from high to low, but it's also going to go faster because it's attracted to that negative opposite charge. So this would occur faster than just typical high-low diffusion of sodium. This is high to low, but also toward a negative charge. This would be faster than that, okay? Pressure can also influence diffusion because if you put pressure on a system, it moves the molecules more, so they're going to diffuse faster. So higher pressure tends to increase the movement of molecules, therefore diffusion. So I just wrote that here. If I put a positive pressure on this side, like a push, like if I'm squeezing on this side or something, that's going to push molecules to diffuse faster than they would normally. Or there's also, we're going to see negative pressure, which is like a pull. If I had negative pressure over here, like you would see in like if I'm pulling like the stopper up a syringe, that creates negative pressure to pull. So we'll see some situations, especially in like plant cells, we'll talk about where there's turgor pressure and that can affect the movement. It'll go faster under higher pressure. Or if there's a positive pressure or a negative pressure, it'll push or pull that molecule to diffuse faster. Facilitated diffusion is when it's diffusing through a protein in the membrane. And we're going to see that's going to be faster. That's at the top of page 3, which comes in later. Surface to volume ratio, we talked about smaller cells have a faster diffusion rate because they have a higher surface area to volume ratio. That's kind of old. And number 8, countercurrent exchange will increase the rate of diffusion And we're going to see that here in the bottom of page 2, which I guess I could just skip to. Let's just scroll down to here's countercurrent exchange, or an example of it. So countercurrent exchange is when there are fluids that are flowing. Let's say we're having diffusion of materials from two fluids that are flowing in opposite directions. And that's going to maximize the rate of diffusion. An example of this we see in the fish gills, which I'm going to show you right here. We also see this in the human kidney, the blood and the filtrate, which is becoming the urine, kind of flow in opposite directions. So it maximizes diffusion of stuff from blood to urine. I won't go into that in detail, but I just want to show you here in the fish gill. So I'm showing here, here's a fish. I think everybody knows fish have gills. They're kind of here behind that little flap. And fish, I don't know if you know this, but their mouths are always kind of moving. They're always kind of pulling water in, and it runs in through their mouth and it runs across their gills and then comes out this little flap on the side. So when you see fish moving their mouths, they're not eating. They're basically breathing. But water, they're just constantly running water across their gills in a one-way direction, in through the mouth, across the gills, and then out. Unlike our breathing where we go in and out. So here's, if I were to look at just like a gill filament, like if I were to take one of these little gill filaments and kind of look at it up close, right? That's what this is sort of showing you. You can see the blood flow. It's going from blue to red here. And just to show you in general, water is coming across the gill in this direction, and the flow of the blood through the gill is going in the opposite direction. So those are two fluids that are moving in opposite directions, and they're going to maximize the rate of diffusion between them. So what's diffusing here? Okay, this is just gas exchange of oxygen and carbon dioxide like we do in our lungs. And oxygen is going to be higher concentrated in the water and lower concentrated in the blood. So oxygen is going to be diffusing in that direction. That's what a fish hopes anyway, that the blood is picking up oxygen. And carbon dioxide is going to be going in the other direction because there's a higher concentration of carbon dioxide in the blood coming from the tissues, and there'll be a lower concentration of carbon dioxide in the water. And so that rate of exchange, let's just look at sort of, why is there going to be a faster rate of oxygen going from water to blood? Let's just look at that. Because these two fluids are flowing in opposite directions. So it gives you kind of two examples here. Again, you can pause if you want to write these down. This is what we're actually seeing. Countercurrent flow, right? We have water going one direction and the blood going the other direction. And this is just showing you percents of oxygen. So it's showing you, that oxygen coming in. Okay, so here's our water coming in. It's going this way. And they're saying it's 100% oxygen. It's not. Water's not 100% oxygen. But let's just say, it's the highest amount of oxygen coming into the gill filament from the water. So let's say if we have a high amount of oxygen here, and you can see as it gets lower as the water flows across, because oxygen is leaving the water and diffusing into the blood. Because there has to be a high to low concentration, because the blood coming in going this direction, okay, the blood coming in has the lowest amount of oxygen because it just came back from the tissues. So it has the 10% and it's gaining 20. As it goes across, you see how this is getting higher because oxygen is being added to it from the water. What you notice, though, is there's always a higher amount, like 100 to 90, 70 to 60, 50 to 40, 30 to 20, 10 to 10, that's equal. But across the whole gill filament here, so in the amount of time that water goes whoosh, and blood is going whoosh, there is a concentration gradient the whole way across. So the diffusion is happening the whole way across the gill filament. So you're going to get a maximum amount of diffusion because these two fluids are going in the opposite direction. Just to kind of make that point clear, this is what would happen if water and blood flowed in the same direction. Let's just look at it. So if the water is coming in with the highest amount of oxygen and it's going this way, and the blood is coming in with the lowest amount of oxygen, it's also going that way. Yes, there's a big concentration gradient, here, and oxygen is going to be diffusing, but there's a time element here. As the water flows and the blood flows, halfway across that gill filament, you reach sort of equilibrium, and so that on the other half of that time, they're in equilibrium, so there's no net movement of oxygen. There would only be net movement of oxygen for the split second, here at the beginning. So we get a greater exchange of oxygen by diffusion from one fluid water to the other fluid blood if they're flowing in opposite directions. That's called countercurrent exchange. And we see that technique a lot has evolved in living things to maximize diffusion. One example is in the fish gills, another is in the kidneys. I just want you to know those examples. Okay, so that's countercurrent exchange, which would also speed up diffusion. So these are all things that would speed up, make diffusion happen faster. So let's look through just real quick at a cell membrane. Here's our cell membrane, our phospholipid bilayer. And we've said this before, but the cell membrane is selectively permeable. So it only lets certain molecules through. Okay, what can and what can't get through. Okay, so I've listed here, based on size and charge, what can and what cannot. What is easily permeable? What can get through? I've outlined in red here. Small, nonpolar, meaning no charge, molecules. Examples: gases, oxygen, carbon dioxide, we were just talking about, can cross cell membranes easily. Nitrogen can also. And then small polar molecules like water, okay, can also cross. So that's kind of what this, if we do a color code here, this picture is sort of showing you, right? We have oxygen, carbon dioxide, gas, so gases, small, non-polar, and we're going to see water, even though it's polar, it's small. Water can always get across, and we're going to see that's going to be a problem. But these things can just squeeze right through the phospholipid bilayer. There's no kind of selectively permeable choice here. These things can just cross without the cell whether the cell wants it to or not, they're going to go high to low. So water and gases can cross whether we want them to or not. Then the things that are not so easy to get across, anything that's large and polar, if it's an ion with like a strong charge, like these examples. And here's an example of large molecules, like organic molecules are large. And so that's what this picture is showing you here. I lost my purple thing. Okay, so this, it's kind of showing you that. So anything that's large, polar or charged, with ions have what we call like a strong charge. Polar is like slight charges. So this mentions water in both. So we're going to see some water can squeeze through, but if we want a lot of water to come in, so let's just cross that out to make it, let's just, for now, assume water can get through. It makes it easier. And then this picture shows you the same thing. And my memory trick, I don't know if you guys remember from freshman year, if you had me. Large, charge, no barge. If it's large or charged, so if it's a charged ion, if it's a large molecule, like an organic molecule, it can't barge. So it can't get through the phospholipid bilayer. Can we move these things across the cell membrane? We can, but they're not going to go by themselves. We're going to have proteins or pumps or channels or specific ways to try to get these in. And these will not cross by themselves. Large charge, no barge. And then the opposite of that is no charge and small can get through. So notice things that are totally hydrophobic, meaning they're small and they have no charge, like gases. They can get through a lot. Things that are small but slightly charged, which means small polar things like water, they can cross but a little, not as much as those. And you can cross out those words. I don't want you to memorize those molecules. Okay. And then things that are large or things that are charged, no barge. So just kind of memorize that. And I think that is where I wanted to end this, because I have another video that now starts here at facilitated diffusion, which is diffusion through a protein. So diffusion is high to low. So I would call this simple diffusion, high concentration to low, but it's going just through the phospholipid bilayer by itself. And these would be only molecules that are small with no charge would do this, simple diffusion through a membrane. And then facilitated diffusion, these two pictures, I'm going to talk about in my next video, are going to be either channels or carrier proteins that can let things come through faster from high to low. Still passive transport because it's going high to low with no energy. Okay, so I'll end there. If I can figure out how to shut this thing off. Oh my gosh.